Well, good morning. We are on the next chapter on fungi, lichen, and mycorrhiza. The fungi, singular fungus, is a group of eukaryotic microorganisms that lack chlorophyll, are unable to synthesize their own food, and are therefore heterotrophic. They live either as saprophytes, obtaining their food from the dead organic matter, or parasites obtaining food from the bodies of living plants and animals. The term fungus is a Latin word meaning mushroom. The branch of science that deals with the study of fungi is called mycology. Greek word mykos for mushroom and logos for discourse. And the branch that deals with the study of fungal disease is called fungal pathology. Clausius, 1601, is regarded as the earliest writers who describe fungi. The founder of mycology is Antonio Micheli. He gave the first systematic account of fungi in his book called Novo Plantarum Genera. General characters of fungi. Fungi is a very large group of over 100,000 species. Fungi are ubiquitous, that is, they are in a variety of habitats. Most of them are moisture-loving and terrestrial, but a few are aquatic, example Monopleparis and Saprolenia, and these are commonly known as water moles. A few fungi are epiphytic, live on trees, etc. Example, armillaria on apple tree and cause a red rot of apple. It is usually defined as a group of those organisms which form the thallus, that is not differentiated into root, stem, and leaves. Built up of single cell or cells, unicellular or multicellular, fungi lack chlorophyll and are unable to synthesize their own food by the process of photosynthesis. Fungi obtain their nutrition from the external source by the process of extracellular digestion and absorption of the digested material. Such mode of nutrition is called heterotrophic, and the organisms are called heterotrophs. Heterotrophic organisms either live on dead, decaying organic matter or on living organisms. According to their mode of nutrition, fungi are of two types, parasites and saprophytes. Parasites may be defined as an organism existing in intimate association with another living organism from which it derives an essential part of the materials for its existence. Thus, these organisms grow in living organisms and obtain their food from it. Their food is derived from it. Parasitic fungi obtain their food from living hosts. These may be ectophytic. These are externally on the host. Example, erysipeae, powdery mildew. Endophytic. These are inside the tissue of plants. Example, albigo, Phytophthora, alternaria. Saprotrophs obtain their food from non-living decaying organic matter such as bread, meat, fruit, vegetables, animal dung, etc. Such mode of nutrition is called saprophytic. Some fungi grow in symbiotic association with the algae to form lichens. Some others grow in close association with roots of higher plants and form mycorrhiza. Fungi possess definite cell wall containing cellulose or chitin or both, that is fungus cellulose, and true nucleus eukaryotic, but lack chlorophyll, achlorophyllous, and differentiation of vascular tissue, that is non-vascular. They are spore-forming and reproduced by vegetative, asexual, and sexual methods. The reserve food materials is in the form of glycogen and oil globules. Fungi grow well at 20 to 30 degrees centigrade and at acidic pH of 6. Fungi may be unicellular yeast, much branched filamentous, rhizopus type. The filamentous thread-like structure that make up the fungal body is called mycelium. Hyphae is the unit structure that make up the mycelium, Greek word for hypha, web. Hypha are of two types, aseptate or sinusoidal, multinucleate, and septate, number of partitions or septa. Aseptate hyphae may form septa during reproduction, example rhizopus, albigo, Phytophthora, etc. In septate form, cell may be monokaryotic, uninucleate, or dikaryotic, two-nucleate, multinucleate. Septa are of three types: complete septum, septum with simple pore, and septum with doliprore. In complete septum, the cross wall is complete without distinct pores, example geotrichum. In most Azomycetes and Dentryomycetes, the septum possesses simple cell pore. Simple cell pore may get plugged by crystalline structure called Warrenin body. Example, Trichomycetes. Worn-in body is secreted by microbody and covered by membrane. In Basidiomycetes, the septum becomes barrel-shaped around a central pore called dolipore septum. It may be surrounded by pore cap. Septopores allow quick transport of nutrients from the region of absorption to all parts of the mycelium. The reproductive hyphae are supplied with the nourishment from the vegetative hyphae throughout the septal pores. When mycelium is interwoven to form web-like structure, it is called placenkyma. It is of two types, prosenchyma and pseudoparenchyma. Prosenchyma is the mycelium in which hyphae are loosely interwoven and lie more or less parallel to each other. Pseudoparenchyma is the mycelium in which the hyphae are very loosely packed, so individuals' hyphae cannot be identified and appear as isodiametric cells, giving the appearance of parenchyma cells in higher plants. When mycelium by interweaving forms compact resting structure, it is called sclerosium. When fungal mycelia are interwoven to form thick cord-like structures like roots that help in absorption, it is called rhizomops. When the plant body is unicellular at one stage and mycelial at the other end, then the organization is described as dimorphic. When the entire mycelium is converted into reproductive structure, the thallus is called holocarpic. When eucarpic forms, only a part of the thallus become reproductive. This can be monocentric, have single sporangium, or polycentric, have many sporangia. The hyphal wall is made up of fungal cellulose, in which cellulose is impregnated with chitin and contains nitrogen in addition to carbon and hydrogen. Chitin is a polymer of N-acetyl-glucosamine. The protoplast is covered by a thin semipermeable plasma membrane. The cytoplasm appears granular and contains many minute nuclei scattered in the peripheral layer. The nuclei are very small and inconspicuous. The cytoplasm contains many small vacuoles filled with cell sap or a number of small gas-filled vacuoles. The vacuoles are small. Few are absent in the actively growing tips of the mycelium. The hyphae are also may be seen to have mitochondria, dictyosomes, Golgi bodies, ribosomes, endoplasmic reticulum, oil drops and glycogen granules, etc. under electron microscope. Reproduction. The fungi reproduce by all the three methods: vegetative, asexual, and sexual. Reproduction. The fungi reproduce by all three methods: vegetative, asexual, and sexual. Vegetative reproduction takes place by various methods as fragmentation, fission, budding, sclerotia, edea, and chlamydospores. In fragmentation, the hyphae of fungus break into small pieces, and each piece may later grow into new mycelium. Fission is common in yeast. The cell divides into daughter cells, which separate by constriction or transverse walls. In budding, daughter bud appears from parent cell after getting the normal size bud break off. When the bud fails to separate after repeated budding from pseudomycelium, edea or round or oval structure having thin walls, the hyphae undergo segmentation and produce yeast-like cells called edea. Each edium on germination produces new mycelium. Some fungi produce chlamydospores, which are thick-walled resting cells. They are intercalary in position. They are capable of forming a new plant on approach of favorable conditions. Gemmae resemble chlamydospores in structure but are not very durable and thick-walled. Many true fungi produce sclerotia, which are hardened, resistant bodies. Sclerotia are capable of surviving unfavorable periods, and as soon as the conditions are favorable, they germinate to produce vegetative hyphae. Asexual reproduction takes place by means of spores. The spores in fungi vary in shape. Spores are usually unicellular, thin-walled, spherical, and diameter ranging from 5 to 50 microns. Several types of spores are reported in fungi. Example: zoospores, sporangiopores, uridospores, teludospores, and pycnospores, etc. Sometimes the spores are produced endogenously in special sac-like asexual reproductive bodies. Pycnospores are small conidal-like bodies produced in flask-shaped cavities called the pycnia or example pycnia. Ascospores are uninucleate, unicellular, non-motile, usually 8 in number, produced in sac-like structure called ashes, characteristic of ascomites. Basidiospores are characteristic of basidiomytes produced exogenously by club-shaped basidium or sterigma. Usually 4 basidiospores are produced. Uridospores and telutospores are binucleate spores produced in clusters called uridosauri. Sexual reproduction in fungi involves 3 processes: plasmogamy, fusion of protoplast; karyogamy, fusion of haploid, 2 haploid nuclei; and meiosis. Fungi may be dioecious, unisexual, some are monoecious or bisexual, some are heterothallic and later are homothallic. Fungi generally possess unicellular sex organs and show gradual degeneration of sexes. Male gametangia is called antheridium. It is smaller in size. Female gametangia is called oogonium, which are comparatively larger in size. The gamete are formed with gametangium. All 3 types of sexual reproduction is present in fungi as isogamy, anisogamy, oogamy. In isogamy, fusing gametes are exactly alike in appearance and function. Fusion of dissimilar gametes is called anisogamy. In anisogamous forms, both fusing male and female gametes are usually motile. Fusion of male gamete with female gamete is called oogamy. In gametangial contact, the 2 gametangia come close together and close to each other but do not fuse. The male gametangium sends a tubular outgrowth called fertilization tube through which the non-motile male gamete or male nucleus migrates into the female gametangium. For example, Phytophthora albugo. In gametangial copulation, 2 gametangia fuse with each other and lose identity in the sexual act, resulting in the formation of zygospore, example muco or rhizopus. In spermatization, some fungi produce numerous minute uninucleate spore-like bodies called spermatia. These are transferred through various agencies to receptor hyphae or trichogyne, a female gametangium. Finally, the contents of spermatium is transferred into receptor hypha through a pore, example Pachynia graminis. In semitogamy, the sex organs are not formed and the fusion occurs between 2 vegetative or somatic cells, resulting in dikariotization, example agaricus. Homothalism is a condition whereby thalli are morphologically and physiologically identical, so that fusion can occur between gametes produced on the same thallus. Heterothalism is the phenomenon in which the fusing gametes belong to 2 genetically distinct strains of the same species, though there may not be any morphological distinction between the gametes of structures bearing them. It was first discovered by Blakesley in 1904. Blakesley found that in muco and rhizopus, certain strains from zygospores, even in pure cultures where different mycelia belongs to the same genetic strain, example Rhizopus sexualis. Heterothalism is a mechanism to perform outbreeding and prevent inbreeding, as it involves sexual reproduction between genetically different strains. The product of sexual reproduction comes to have different alleles and different genes linkages. It introduces variations that are helpful in adapting to diverse habitats, unfavorable environments, and toxic chemicals. The classification of fungi. Fungi are divided into 4 classes according to the septate or septation of the mycelium and on the basis of characteristic features of reproduction. Phycomycetes, Aspermycetes, Basidiomycetes, Dentryomycetes. Phycomycetes are algae-like fungi. Hyphae of Phycomycetes are sinusitic and non-septate. Phycomycetes are entirely aquatic and known as water moles. Phycomycetes are the most primitive true fungi. In Phycomycetes, reproduction takes place by both sexual and asexual methods. Asexual reproduction takes place by pores, spores. Motile spores are called zoospores and non-motile spores are called eplanospores. Chlamydia spores are those thick-walled resting spores which are formed on the hyphae of some lower fungi. 2 types of flagella are present In Phycomycetes, these are whiplash and tinsel types. Sexual reproduction in Phycomycetes take place by planogametic copulation, gametangial contact, or gametangial copulation. The most common examples of Phycomycetes are Saprolina, Rhizopus, Muco, etc. Other classification of fungi based on the features. Common name: mycelium, asexual reproduction, sexual reproduction, and the fruiting body. We have Phycomycetes, which are algae-like fungi, aseptate, cenocytic, zoospores, aplanospores, chlamydospores, sporangiospores, isogamy, oogamy, and zygospore. Asomycetes, sac fungi, septate, branched, unicellular, conidia, budding, fusion of compatible nuclei, ecospores, asospores formed in ascus. Esocarp, let me get that word clear, claystot, that's claystot, Technium, Parathecium, and Apothecium. Basidiomytes, club fungi, secondary mycelium eukaryotic, Edia and basidiospores, somatogamy, blasidiospores formed on steric matter, basidiocarp. Fungi imperfecti, branched septate mycelium, conidia, absent or not known, absent. Asomites are common fungi, commonly called as sac fungi. Asomites are characterized by well-developed thallus and production of asospores. Cell in asomites are uninucleate or multinucleate. Mycelium of asomycetes are branched and septate except in yeast. Yeast is unicellular. Asomycetes, asospores are formed inside the ascus during the sexual reproduction. The typical number of asospores in an ascus is 8. No flagellated cells are found in asomycetes. Asexual reproduction may take place by budding, fission, fragmentation, edia, conidia, formation, etc. Sexual reproduction takes place by plasmogamy, which may be isogamous or heterogamous. Fruiting body of asomycetes is asocarp. Asocarp may be clastothecium, globose having no natural opening, parathecium, globose having no apical opening, or apothecium saucer-shaped. The most common examples of asomycetes are penicillin, neurospora, yeast, etc. Basidiomycetes. These are commonly called as club fungi. Basidiomycetes resemble the asomites in having septate mycelium production of non-motile spores. Dicaryotization or diplodization is the process by which a binucleated condition is attained from uninucleated conditions. Basidiomycetes have a short-lived uninucleate state and a dominant binucleate stage. Cells of basidiomycetes are made up of chitin, mannan. Basidiomycetes are characterized by the delipore, septa, and clamp connections. A hook-like clamp is formed which help in passage of nuclei in the mycelium. Basidiospore are formed over the basidium during sexual reproduction. Fruiting body of basidiomycetes is known as basidiocarp. Asexual reproduction occurs by fragmentation by means of spores such as conidia, edia, arthrospores, etc. No specialized sex organs are present in basidiomycetes. The life cycle of basidiomycetes consists of 3 clear and distinguishable phases. These are plasmogamy, karyogamy, and meiosis. Common basidiomycetes in Eustylago, Pycninia, Agaricus, Polyporus, etc. Dentryomycetes is an artificial group without any common relationship. Dentryomycetes are commonly called as fungi imperfecti due to the absence of perfected sexual stage. Dentryomycetes have separate hyphae and reproduce asexually by means of conidia. Beside conidia, thalasspores are also found on some fungi imperfecti. 2 types of thalasspores are chlamydospores and arthrospores. Common dentryomycetes are cercospora, Galactotrichum, Pyracularia, Fusarium. Harmful and beneficial aspects of fungi. Some fungi are used as delicious food. The fructifications of certain fungi are used as nutritious and delicious food. Example, Agaricus bisporus and Acampestris mushrooms, Mochella and Lycoperdon, Clavacia, Pleurotus, Bovaria, Wolveriella, etc. are also edible fungi. Yeast is an important source of vitamin B and D. Saccharomyces, Endomyces, Rhodotorula, Torulopsis are rich in protein. A food called sufu is produced from mucor and antimucor. Fungi are used in the production of differential antibiotics, substances of microbial origin, and have antimicrobial activities. In alcoholic industry, yeast, Saccharomyces, is used for fermentation. In baking industry, CO2 evolved raises dough. Enzymes like teca-diastase, digestin, and polyzyme are produced from Aspergillus flavus oryzae series. Invertase is prepared from Saccharomyces, Cerevisiae, amylase from Aspergillus oryzae, zymase from yeast, Saccharomyces, Cerevisiae, Penicillium camemberti, and P. roqueforti are used for flavoring cheese. Various organic acids are obtained from fungi. For example, citric acid by Aspergillus niger and A. wendtii, muco, etc., gluconic acid by Aspergillus niger and Penicillium pepogenium, gallic acid by Glaucum and Gallomyces, kojic acid by A. oryzae, fumaric acid by Rhizopus stolonifera, gibberellic acid is obtained from fungus Fusarium monoruliforme, Gibberella fugicori. Fungus widely used in genetic engineering is Neurospora crassa. For working on Neurospora crassa, Beadle and Tatum, 1941, proposed one gene, one enzyme hypothesis and received the Nobel Prize in 1958. Aspergillus niger is widely used to determine available copper, magnesium, potassium, and molybdenum in the soil. Saprophytic fungi live upon dead organic matter and thus break down complex substances into simple ones which are again absorbed by plants and has increasing soil fertility. Soil inhabiting fungus Trichoderma kills Pythium. Fungus, rootroid fungus, similarly penicillin, vermiculatum, Shex rhizotonia solani. Predaceous fungi like Dactylella, Dactylaria, Zoophagus, and Arthrobotrys, for example, destroy certain nematodes, eelworms, etc., causing plant diseases. They help in absorption of water and nutrients and in turn get ready-made food, symbiosis. Absidia, Aspergillus, Cladosporium, Mucor, Penicillium, and Rhizopus have soil-binding property, but mucilage and they make the soil good. Many insect pests can be controlled by using fungi like Ashsonia, Elyroides, Asoria farinosa, Impusia, etc. Few fungi like Saccharomyces and Rhodotorula fix atmospheric nitrogen in soil. Fungi is the causative organism of many diseases in plants, animals, as well as in humans. Claviceps purpurea cause ergotism and also yield a hallucinogenic drug called LSD. The potato crop was destroyed in Ireland in the middle 19th century due to infection of Phytophthora, disease associated with fungi. Plant diseases: blackpot disease of potatoes, Syncytium endobioticum, white rust of crucifers, Albugo candida, Cystopus candidus. Late blight of potato, Phytophthora infestans. Famous famine of Ireland, 1845, is associated with this disease, which caused death of lakhs of people. Early blight of potato, Alternaria solani, powdery mildews, Ericipe sps. Powdery mildew, Ericipe sps, damping of seedling, Pythium sps, loose mud of wheat, Ustulago fitici, black rust of wheat, Buccinia graminis fitici, apple scab, Venturia inequalis. The black rust of wheat, Buccinia graminis fitici, apple scab is Venturia inequalis, downy mildew is Peronospora sps, red rot of sugarcane, Calypso trichum falcatum, ticker disease of groundnut, Cercospora sps, stem called of coriander, Protomyces sps, wilts of Arar, Spisurium sps, leaf rust of coffee, Emilia Bestatrix, blast disease of rice is Paracularia oryzae, Greenia disease of bajra is Sclerospora graminicola, flag smut of wheat is Urosystidus tritizae, maize smut is Eustelagomatis. The loose smut of oat is Eustelagoabanae, covered smut of oat is Eustelagocolerae, covered smut of jova is Sorghum Sphacilotheca sorghai. Severe famine in Bengal 1943, which caused death of a large number of people, was due to this disease. The human diseases: aspergillosis, lung disease, Aspergillus niger, A. flavus, A. fumigatus, ringworm, Trichophthalon, Microsporum, Neuritis, infection in nervous system, Mucus pusillus, mental disorder, Cryptococcuses, Lipomyces neoformans, ear infection, Otomycosis, Aspergillus flavus, A. nigilans, thrushwater throat, Monilia, animal diseases, athlete's foot, Biotinia rubrum, ringworm, Trichophyton, Microsporum, Epidermophyton, Mucormycosis Rhizopus, Aspergillus, Pencilosus, Penicillium. Belatus and Amanita spp are poisonous fungi. The fungi that produces latex is Mycena. Mycorrhiza, Penicillium, Neurospora, Amanita, polyporous are commonly called a spin, bread mold, green mold, pink bread mold, toadstools, and bracket fungi respectively. Spores of Rhizopus, Mucor, Aspergillus germinate on jam, jellies, pickles, bread, etc. and destroy them. Penicillium, Mucor, and Aspergillus destroy meat. Poroporous, pore fungi causes wood rot. Alternaria, Penicillium, Trichoderma, Mucor, Ketonium, Cephalothecium, Fusarium, destroy leather, cloth, rubber, paper, camera lenses, etc. Aflatoxin or mycotoxins, harmful secretions produced by Aspergillus flavus, A. fumigatus, Penicillium islandicum, etc. These bind with the DNA and prevent transcription, hence protein synthesis. These cause liver cancer in animals and men. In VAM, the hyphae develop an arbuscule penetrate within the cortex of root. It helps mainly in phosphate absorption from the soil. Lichens. Lichens are formed by a symbiotic relationship between algae or cyanobacteria and fungi, in which individual photobind cells are embedded in a complex of fungal tissue. The number of lichen genera is 400 and species over 15,000. The study of lichens is called lichenology. Body of the lichen is made of a fungus partner called microbiont and a photosynthetic algal partner called photobiont or phycobiont. In 98% of the lichens, the microbiont or fungal partners belong to Asomycetes. Few lichens shows Basidiomycetes fungal partners also. Photobionts or phycobionts either belongs to cyanobacteria or green algae. Photobiont performs photosynthesis, nitrogen fixation, elaborates vitamins and other growth substances like microbiont takes part in protecting covered body, a protective covering body, attachment, absorption and retention of moisture from dew, rain and wet air, and protection against harmful radiations. History of lichen. The term lichen was first given by Theophrastus for superficial growth on Bacchabilia europea olive tree. Morris in 1699 called lichen as Muscofungus. Schwendiner, 1867, gave dual hypothesis and established the composite nature of lichen. He defined lichen as fungi parasitizing algae. Bonnier, 1886 to 1889, successfully synthesized a lichen by growing fungal spores with algae. Ray Inninke, 1872, gave the term consortium for the association of algae and fungi. The term means mutual growth and interdependence. DeParry, 1879, termed this association as symbiosis. Krombeck, 1885, gave the term Helotism, that is a master and slave relationship to the algal and fungal association lichen. Helotism is the most accepted term used nowadays to describe this relationship. Algae are present on the upper part of thallus and fungi are present on the lower part of thallus. Fungal partner provides protection and are also responsible for the sexual reproduction. Such a mutually beneficial relationship is called symbiosis or mutualism. For proper diffusion of nutrients, the algal cells and fungal hyphal tips become surrounded by common extracellular substance. At times, the microbiome send haustoria into algal cells, prevent alga to secrete pectic substance, or induce alga to secrete nutrients. Consequently, fungus is considered to be controlled by parasite. The phenomenon of controlled parasitism is called helotism, as algae and fungi both contain cell wall, which is the characteristic feature of plant cell, so lichens are considered as plants. About 12 genera of cyanobacteria and 21 genera of green algae are considered as lichen symbiont. Important cyanobacteria are nostoc, Gliocaspar, and Ribularia. Green algae symbiont involves Protococcus, Centrophilia, Cladophora, and Triboxia. Lichens generally grow on old walls, roof of house, trunks of trees, or exposed rocks. Lichens like Usnea hangs from the smaller branches of trees. Lichen can withstand extreme of cold, heat, and drought. They are dominant form of vegetation in alpine and arctic tundras. Reproduction may be vegetative, asexual, or sexual. Vegetative reproduction occurs by fragmentation, when thallus breaks into small fragments, develop into new lichen-thalli. Soredia are small masses of hyphae enclosing a few algal cells. Soredia are dispersed by acorns. Each sodium develops into new thallus. Aicedia, they are superficial outgrowths from the upper surface of the thallus. Aicedia consist of an external cortical layer and an internal algal layer. Asexual reproduction. Some lichens, pycnidiospores, are produced at tips of fertile hyphae inside pycnidium, which is a flask-shaped structure, opening through a pore called osteo. These spores germinate to produce a fungi mycelium. The fungal mycelium walls corresponding alga form new lichen thallus. Sexual reproduction. It is formed mainly by its fungal component. Most of the lichens belong to the division Ascolichens, which the fungal partners belong to Ascomycetes. The male reproductive organ is flask-shaped. Spermagonium, which produces non-motile male gametes, is spermatia. The female reproductive organ is Scarpogonium, which is differentiated into basal called Ascogonium and upper long tube like Trichogyne. The fruiting body ascocarp formed out of fertilization may be apothecium type or perithecium type. The bottom ascocarp is lined by hymenium, which consists of asci and spread with paraphysis. The wall of asocarp, apothecium or perithecium, is composed of the vegetative walls of the thallus consisting of algal and fungal layers. Each ascus contains 8 ascospores which germinate and form fungal mycelium. The mycelium, when it comes in contact with a suitable algae, forms a new lichen thallus. Classification of lichens. On the basis of the fungal partner, the lichens are divided into 2 groups. Esolichens, fungal partners belong to Esomycetes. Gymnocarpae, fruiting body is apothecium type. Parenocarpae, fruiting body is perithecium type. Basidiolichens, fungal partners, belongs to Basidiomycetes. On the basis of habitat, lichens are saxicolous, rock dwellers, and adaptation is xerophytic adaptation, example Xanthoria. Corticolous, bark dwellers, and grows in adaptation of plenty of marsh, example Amelia. Terricolous, terrestrial species and thus grows in soil, example Cladonia, Lurkinia. Lignicolous, lichens which grow on wood directly, example Cyphelium. On the basis of thallus, lichens may be leprous lichens. The lichens are in the form of minute scales attached superficially over the substratum, example Lepraria incana. Crustose, lichens are crust-like, closely attached to substratum due to adhesion of several points, example graphis, rhizocarpon. Foliose, lichen body is like a crinkled and twisted leaf, example flat, branched or lobed. It is attached to substratum by one of a few points, example Parmelia and Peltigera. Fruticose, the lichen shows branched, erect, or pendulous with bushy appearance. An attaching disc is present at the base, example Ramalina, Cladonia usnea. Filamentous, the photosynthetic partner is well developed. Filamentous is discovered by a few fungal hyphae, example Rhacodium. On the basis of distribution of algal component in the thallus, lichens are of two types. Moisomerous thalli, algal cells and fungal hyphae are uniformly dispersed throughout the thallus called colyme. Heteromerous thalli, the algal cells are found in algal zone only. The bulk of lichen body is formed by fungal partners, microbiont. It includes the surface, medulla, and rhizinae. The algal constitutes hardly 5% of the lichen body, example Parmelia. Economic importance of lichens. Freshwater species of lichen is Hammonelia lacustris, and marine water species is Caloplaca marina. Cladonia rengiferina, commonly called as reindeer moss, is an important source of food for reindeer. Certeria islandica, commonly called Iceland moss, is used as a source of food for sheep, cattle, as well as humans. Everina used for making bread. Leucomora is also edible. Lichen, Evernia prunus tree yields an excellent perfume. Rachella, Parmelia, Evernia, etc. are dye-yielding orchi lichen species. Litmus is obtained from Lichen, Rachella as Montagne. Medicinal property of lichens is due to a substance lichen. Usonic acid, an important broad-spectrum antibiotic, is obtained from Usnea and Cladonia. Xanthoria parietina is used against jaundice. Peltigera canina is used against hydrophobia. Certeria is used as a laxative. The protolycasterinic acid obtained from some lichen like Certeria icelandica has anticarcinogenic properties. Lichens serve as indicator of air pollution, as they are very sensitive to air pollution, especially SO2 pollution. Crustose lichens are pioneers in xeros, share, succession beginning in dry condition. In Russia and Sweden, some lichens like Crataria and Leuconoria are used for alcoholic fermentation. Lichens secrete some organic acids which break down rocks and thus help in soil formation. Some lichens are poisonous also due to various substances present in them, as lichen, Lithera vulpina, vulpinic acid, Citreria juniperinina, pinastrinic acid, Parmelia molluscula, selenium, Xanthoria parietina, beryllium, Ebrina farfusiae is chlorine. Mycorrhiza is a symbiotic relationship between fungi and roots of higher plants. They are thick, irregular, woody covering devoid of root hair and root cap. In a mycorrhiza association, the fungus may colonize the roots of a host plant either intracellularly or extracellular. This mutualistic association provides the fungus with a renewable source of food through access to fixed carbon sugars from the plant photosynthate. These are translocated to the root tissues from their source locations, usually leaves, and then to the fungal partners. In return, the plant gains to use the mycelium as tremendous surface area to absorb mineral nutrients from soil. The mycelium mycorrhiza networks have better mineral absorption capabilities compared to plant roots. Mycorrhizal plants are more resistant to diseases such as those caused by microbial soil-borne pathogens, and also more resistant to the effects of drought, perhaps due to the improved water uptake capability of the fungal hyphae. Types of mycorrhiza. The two most common types of mycorrhizas are the ectomycorrhiza and endomycorrhiza, more commonly known as arbuscular mycorrhizas. The two groups are differentiated by the fact that the hyphae of ectomycorrhizal fungus or fungi do not penetrate the cell wall of the plant's root cells, while the hyphae of arbuscular mycorrhizal fungi penetrate the cell wall. Ectomycorrhizae typically form between the roots of woody plants, and fungi belong to the divisions Basidiomycota, Acimycota, or Zygomycota. These are external mycorrhizas that form a cover on root surfaces and between the roots cortical cells, between the mantle formed by the mycorrhizae. Most of the biomass of the fungus is found branching into the soil with some external to the apoplast stopping short of the endodermis. These are found in 10% of plant families, mostly the woody species, including the oak, pine, eucalyptus, dipterocarp, and olive families. Endomycorrhizae, arbuscular mycorrhizas, or VAM, formerly known as vesicular arbuscular mycorrhizas, involves entry of the hyphae into the plant wall cells, cell walls to produce structures that are either balloon-like vesicles or dichotomously branching in machinations of buscules. In VAM, the hyphae develop an arbuscule penetrate within the cortex of root. It helps mainly in phosphate absorption from the soil. The fungus is generally a Zygomycetes. The fungal hyphae do not, in fact, penetrate the protoplast, that is the interior of the cell, but invaginate the cell membrane. The structure of the arbuscules greatly increases the contact surface area between the hyphae and the cell cytoplasm to facilitate the transfer of nutrients between them. Arbuscular mycorrhizas are formed only by fungi in the division Glomeromycota, which are typically associated with the roots of herbaceous plants, but may be associated with woody plants. Arbuscular mycorrhizas are likely to be very helpful in protecting plants from adverse conditions such as lack of water and nutrients. Arbuscular mycorrhizal fungi are quite extraordinary organisms. First, they have been asexual for many million years, and secondly, individuals can contain many genetically different nuclei, a phenomenon called heterokaryosis. This type of association is found in 85% of all plant families in the wild, including many crop species such as the grains. For more mycorrhiza, refer chapter on pesticides and biofertilizers. That is the end of this chapter. Please listen well, read well, make your notes, take adequate screenshots, and prepare well for the exams. We have a Q&A at the end of all the session on each week or each month as well, each month end as well. So prepare well. I wish you all the best. If any questions, any doubts, queries, or clarifications, please feel free to come onto the comment box, or you can connect on my WhatsApp. Just please send in queries, because I cannot be convenient enough, or I wouldn't have the convenience to attend calls, because I am Truecaller monitored, and too many calls would only jumble up and cause a lot of clutter. So I wish you all the best. Study well, do well. To connect for any doubts that you may have, right? So I'll take leave. I'll be going into the next chapter in a couple of moments from now. See you then. Bye.